Hi everyone, I hope you had a nice spring break. It's my pleasure to introduce today our next speaker, Professor Hallatschek. He was born in Germany. He completed his doctorate in Berlin after studying in Heidelberg and Zurich, and he then pursued a postdoctoral research at Harvard before leading to the Max Planck's research group on biology and biological physics and evolutionary dynamics in cognition. Since 2013, he has been a faculty member at UC Berkeley, where he holds an appointment in both physics and integrative biology departments. His work has earned numerous accolades, including the Science Investigation Award, the NSF Career Award, and the prestigious Humboldt Professorship. Please join me in welcoming Professor Hallatschek.
Well, welcome everyone. Thank you so much for inviting me to come here and to be back in Boston. I always enjoy being here, even though my postdoc was in the main campus. So I rarely got to go to the medical department. My lab is very interdisciplinary. We say we are hybrids. We do both experiments and theory. My background is in theory, but we also do kind of wet lab work with microbes, bacteria, and yeast to study evolutionary dynamics. And so the picture in the background is one that represents a so-called Muller plot. So each sliver here represents a different variant in a population. And the frequencies or fractions of each of these variants, they have to add up to 1 at every time point. So x would be time here. And as a consequence, you see this changes in abundance or frequencies of the different variants. And if a variant would be spreading, it would be kind of potentially exponentially growing. And taking over the population. If it's shrinking, that's the opposite. And what you see here is mostly neutral. But we have looked at such plots in our lab by studying actually test tube populations of bacteria who carry genetic barcodes. And we use these barcodes to trace lineages within these populations. And by just analyzing the resulting Muller plots, so to speak, we could learn quite a bit about evolution, about mutations in these populations. But we developed computational techniques to do so. And when then, COVID was in full swing, we realized, oh, there's lots of data like that also arising in COVID. And so maybe we can apply some of this to COVID. And there actually new challenges arose, but also opportunities, because you get then sampling different locations, which allows us, and this will be the main point here, to actually infer how cases were reported at what rates between different regions. So let me get to that after first a bit of motivation. So if you think about modeling a phenomenon or entity that is spreading over time, then one often writes down differential equations, maybe stochastic differential equations, that lead to the growth of that entity. In the simplest case, where you ignore space, you say it's populations like well-mixed test tube, then just schematically, the way you would write down such an equation can look like this, where x of t would be that entity that is growing, and the rate of change of that entity is controlled by growth, where r of t is the current growth rate, which of course can again depend in some complicated way on what x currently is. And then I added here a second term, which has to be there always, which is a noise term that characterizes how stochastic transmission is, how stochastic reproduction is. It's called here demographic noise. In the population genetics community, it's called genetic drift. And with that, you can maybe try to model something like just overall abundances over time. But often we are interested in not only the overall abundances, or even to predict them, we need to often resolve space. And then you have to come up with more complicated models where, here I'm depicting a meta population model where you say, okay, we can subdivide the population into different themes, these little circles here. And these themes, they import cases, right? There's dispersal between the different themes. And so I have to introduce a matrix, lambda ij, I'll say more about this matrix later on, that somehow characterizes how infections are being carried around. And then a more general model would look like this, where you have the same terms as before, but in addition, a term that describes how cases are brought to deem I here from other deems. Okay. And maybe with that, then we can study the spread or predict the spread of epidemic or something else, beneficial mutation in space and time. But of course, to actually do this, we need good parameters. To actually predict, to come up with good models, we need good parameters. And people correctly obsessed during COVID to think about growth. Like R naught, everybody now knows what R naught is, and it was very important to figure out what that is. But it can be much more challenging to get good estimates or even measurements of these other parameters, like demographic noise or how cases are being imported. And so to do this, one way is perhaps, sorry, I'm going too fast here, go back. One way to do this is to look, for example, if you're interested in dispersal, you could look at mobility data, how people are being moving around. But that's not everything that matters here, because clearly the way cases are being imported from one region to another depends not on, well, you certainly need to have mobility, but it also depends on the behavior of the people, on the infection levels, right? It's a compound parameter that's very, very hard to estimate. And so what's, I would say, urgently needed would be kind of direct measures, direct ways of getting at these parameters. And as far as we know, they are very little. And so we try to, at least retrospectively, get a better view of these two parameter sets using, these kind of fluctuating time series that I showed you before. So what kind of data we really used? So one of these papers is already published, and the other one is a preprint at this point, being in a very, very long review process. So we focused on England in both cases simply because it was sequenced extremely well, especially during 2021. And another reason to focus on England was because, we had this discussion before, strangely but conveniently, it had like very high abundance levels, especially during the Delta variant, which were kind of constant, kind of a plateau phase, where you wonder, why are the infection levels high but constant, right? You would think R naught is larger than 1, then it should grow, or smaller than 1, then it should shrink. Why is it stable? There are hypotheses, and I don't want to go into that. I just want to say, for us, it's very convenient because both the communications were, this is kind of from an infection survey, which randomly sampled people, were constant, and the sequences that were sampled were also roughly constant during that time. So we are going to focus on that period of time. And then extract from this period the different variants we can see. So here you see Mahler plots for, I don't know why it's shaking. Is there an earthquake? California, you always have to worry about that. So there are, of course, different, big variants, and there are some selective differences between them. But if you look into the delta variant or into the alpha variant or BP1177, you find lots of practically neutral variants where you see here, okay, what you see is, what you mostly see are fluctuations. And so our question was, what can we learn from these fluctuations? And the hypothesis is we can learn about these two parameters that I mentioned before. The kind of obvious thing to get at is to actually measure the biological fluctuations of the transmission process that is happening from fluctuations here. And you can, even just by looking at this plot, you might say, oh, the fluctuations are large initially, and then they become smaller and smaller. Maybe that really reflects the fact that the population size of delta, of course, was increasing, and that tends to go together with less fluctuations. Okay, and this is Shinshin right there, who was really leading this process. I was trying to, how can I embarrass you during this stuff? I'm not really embarrassing you at this point. Maybe there are other opportunities later. She was really spearheading this project, and so here you see again the kind of the data that we looked at, and the main challenge was this: that the reasons for the fluctuations that you see here are twofold. One is genetic drift, which is really the biological noise we're interested in, right? So there is just due to stochasticity and the reproduction and transmission process of the virus. You expect that true frequencies to change from one time point to the next. And the variance is expected to have this form where NE is a parameter that is called in his population genetics. It's kind of a famous parameter. It's called the effective population size. It quantifies the level of fluctuations, and in the simplest possible models, the effective population size should be identical to the census population size, which it often isn't, and also in this case, we see it is not. But there's a second source of fluctuations, namely, we don't see the true frequencies of these variants. They fluctuate just because of observational noise. You are measuring these frequencies by going into the population, sampling individuals, and then based on the sample, you determine the frequency. So just there will be chance effects in your measurements that actually also scale, the variance scales linear with the frequency. The scaling with F is here identical. But there's a big difference between the two, which is something we are going to exploit, namely the kind of per generation errors that are introduced by genetic drift. They compound over time, they add up. But the observational noise is really just kind of fake noise that, if you have it in one generation, this noise will go away in the next generation. There will actually tend to be anti-correlations. And most importantly, it doesn't add up over time. And so the contrast between the two is something we exploited in a computational method that Shinshin developed in order to then infer genetic drift. The method is based on a Markov model. It's one that is very similar to the second project where I go more into the details. So I don't want to describe it here. I just want to give you the take-home message. Namely, it is that the effective population size that Shinshin inferred was much smaller than expected, based on the actual abundance of community cases in England, and by a factor of 100 or even more. And correct me if I'm wrong, Shinshin. I think that's what we found, and that is also, that cannot be explained by the impact of just individual superspreaders, so people that would be superspreaders, but it can be explained, and this is really potentially just one explanation. It can be explained if you think there is an underlying spatial structure where you have kind of continuously happening sub-outbreaks in different parts of the population. So just imagine that the population can be decomposed into cliques of certain sizes. Imagine just your bigger circle of friends, and then you can imagine that you're better connected with your friends than with other cliques. So there's strong connections within these cliques, weaker connections between cliques. In such a situation, you can imagine that locally within a clique, if you don't know anyone who has COVID right now, maybe you behave less careful. Therefore, practically within your clique, you are not as larger than one. So if the pathogen hits that clique, it's going to spread locally exponentially for a while until the clique has changed its behavior and then kind of goes away. And if that's happening over time, you have these exponential sub-outbreaks, which lead to a lot of noise in this pattern, and an effective reduction in the population size by the size of these cliques. Because essentially what then matters is not how many individuals are in the population, but how many cliques are in the population. So this factor of 100 or more would then be roughly the size of these cliques. By the way, if you have any question, don't wait until the end. I much prefer if you ask questions during the talk. We have enough time, and I have too few slides. So this is what we had published here. We have actually, there's some additional piece of data, which to me is suggestive that this might be going on. But it's really just an hypothesis, right? What we are offering here. And there might be others that explain this reduction. But there's one piece of evidence which to me strongly suggests that there are exponential sub-outbreaks. And that has to do with the following kind of data. So Takashi, who was a collaborator here, is featured more on the next slides. He looked at the mutations that really arose in this plateau phase. So that didn't exist before. They arose in the plateau phase. And then asked, what is the frequency spectrum of these newly arisen mutations? And if really the dynamics here where the overall abundance stay constant, so that there's no expansion, no shrinkage, you would expect that these neutral mutations that appear in this plateau phase, they would satisfy the spectrum corresponding to neutral mutations. That would be the blue dash line here, which is expected according to the Red-Fisher model. But in fact, the site frequency spectrum has a very different behavior, which is consistent with the existence of so-called jackpot events. This is a statistic that goes back to Luria and Delbert, very old study of number of colonies on plates, essentially as a measure of the clone size distribution of resistant mutations that appear in overnight cultures. They were also characterized by these jackpot events, and we think we have evidence that they exist in this plateau phase too. But we need to more carefully analyze this, and this is still unpublished. Any question? Yeah.
So this
Maybe can we use the mic?
So I see you've got there the set frequency spectrum is predicted by a Boltzmann columns, and presumably the ancestral process for this, however, doesn't correspond to it exactly. Have you looked into what it would?
Say it again. It
What
doesn't.
would the ancestral process look like for these sort of for this sort of a clique model? Because I see you've got a spectrum of Boltzmann coalescence there.
Yeah, this is really just.
You've got a divergence there already going on.
Yeah.
So
No, this is only the low frequency part of the spectrum, because you can imagine the new mutation that arise here, they tend to be low frequency, so you don't have the high frequency tail. For the Boltzmann coalescence, there's really the high frequency tails tells you something about the deep coalescence.
yes.
And there's something very interesting going on there as well. We don't expect the deep coalescences to be characterized by the exact same demographic structure. But what this initial decay nevertheless is about these exponential suboptimals. This is exactly what you expect. The questions? There.
Oh, very good. So is the idea here that you have like all of the delta sort of like data, and then when you calculate the effective population size with your method, like you find that it's less, and so that's how you infer that you have these sub like.
Yeah, it's just
Okay, cool. So the question is like with smaller population sizes, usually if I remember correctly, you have like higher drift. So how do those, if you have these subpopulations, do you have higher drift then there? And then if so, like how does that explain the sort of like neutral behavior that you see?
yes, so really very, very roughly speaking, right? What's going on is that each clique acts like just one individual infected person. Like if you say, oh, normally this should be the total population size, right? We also say, okay, it's a kind of coarse-grained picture. We don't describe individuals as consisting of many cells, right? We just say this is one individual and it can be either fully infected or not infected. We don't make a difference. And here we say like kind of the excess in fluctuations is as if the cliques are either all infected or not infected. We have now a tiny bit better measurement, this high frequency spectrum that captures also kind of the in-between cases, suggesting their exponential sub-outbreaks within these cliques. Sure. Others? All right, so then to go on, we realized, well, here we actually lumped together data for which we had location labels. And so we thought, can we do more with these time series of just neutral variants using the location labels? And turns out, yes, you can. And I'd love to have even more labels than just locations, different population groups. Because it turns out you can learn about how cases are being imported between population groups if you have those labels. And I want on this slide just to give you the very basic idea of where the signal is coming from. And then it becomes a bit more mathematical, but it's all going back to this very basic idea. And this was now spearheaded by Takashi, who's now professor in Kyoto. And so to think about this, imagine we have just two subpopulations, A and B, and they are at first in a lockdown phase, so they don't exchange any infected individuals up until a certain point in time. And then I want you to imagine a neutral variant that by chance is at high abundance initially in population A and at low abundance in population B, right? In fact, there might be, look at many variants, so we might have many variants that satisfy this. So in the background, it's very hard to see there are here these slightly, so the dashed line is really an average over many realizations that you see here in the background. And this is just to say that, in the absence of any exchange between A and B, these frequencies, of course, have no reason to become more similar over time. They stay separate, they fluctuate around their initial values, but that's it. But then when you imagine lifting this lockdown and they start to exchange migrants, what's going to happen? Well, what's going to happen is they become more similar because of that exchange of migrants, but also the way they become more similar reflects who's importing more cases from the others. Right, so here we, in this simulation, we just envisioned that A is importing more individuals from B than the other way around. And then the consequence is that the frequency in A becomes more similar to how B was than the other way around. So this process. So by looking at the dynamics of what these frequencies are doing in different regions and correlating them, maybe we have a chance of getting at these arrows from flotation rates, and even in a kind of directed manner. And that's indeed a signal that you can see in, this is really just, if you think about modeling this, what's happening here with these frequency variables, they actually are very similar, for those who care, like Brownian particles in a harmonic trap. The Brownian particles are diffusing, these are the fluctuations you see in the background, you turn on the harmonic trap, and then it gets attracted to the center of this harmonic trap, and the trap stiffness is something about case importation rates. This is really not key to understanding the rest. I just wanted to illustrate here that you can really see this regression to a common mean if you look at individual time series of just SNPs. This is what you see here. These are just individual SNPs, two SNPs, for which you see the frequency time series during the Delta variant in different regions of England. So here the different colors correspond to regions. I mean, somehow England, somebody decided to divide it up into 9 regions. And those 9 regions are shown here, funny names, and abbreviated here. And you see how indeed the frequencies become more similar over time. Of course, there's a lot of noise, but the power of this method arises when you average over all possible SNPs to see this convergence. And in fact, here's another just empirical measure. It's the autocorrelation of different regions I and J by where you do the average over all possible SNPs, and you see indeed a decay of these correlations with time. And it also depends on distance between regions, which is something I'm going to go more into later. Question, this problem, which is really just, I mean, that's kind of important. I think that is still understandable. Hope this is understandable because this is where the signal is.
So can you just tell us about one point on there to make sure, I think I know what you're saying, but I'm not positive. So
Yeah.
Tell us, interpret the rate point that's at 0.5. What exactly is that thing?
So the red line, see the red line? Oh,
Or just like one point on the line?
yeah, so just draw it. So it's kind of asking how correlated are Rij looks at the frequency difference between of a given variant between regions I and J, averaged over all variants.
And when you say variant, you mean SNPs.
In this case, exactly,
Yeah,
SNPs
right.
and neutral SNPs.
Yes, right, yeah.
And it's a function of time because we are looking at how this frequency difference changes over time. So we're looking at the frequency time difference at a later time correlated to an earlier time. These correlations decay because of this regression to some common mean. And what this shows not only the decay, it shows actually that the decay is more rapid between a region that is close by, like London and Southeast, sorry. And then blue and green are 2 regions, London and North England, and London and Yorkshire and the Hunter, which both show us lower decay, which is kind of consistent with, well, they are further apart, so have less, the propagation rates are lower, and this regression should be slow.
So I think the thing I'm having a hard time following, I would have thought that at time 0, if you just take frequencies in one place and at another in place I and place J, it wouldn't be perfectly correlated.
Yeah, it's not, sorry, it's normalized to be one.
Normalized to be.
Yeah.
I see. Okay.
Yeah.
You only
Throw it to her. I love to throw these things, by the way.
Okay. We only take neutral SNPs that have distinct geographic patterns, so
Sorry,
like,
say it again.
do you only take neutral SNPs that have distinct geographic signatures, so like that is high in one and one in the other, or just
No,
all neutrals?
all of them.
Okay.
They. So one thing that's nice, let me say it now because I might forget it. For this method, what is really useful is that so the method works really well once you have a lot of data. Naturally, you get a lot of data once a variant is highly abundant. And at this initial time, the variant went through an explosive phase where actually the fluctuations were very, very large. And the consequence of that is that directly after this initial big bang that happened for a variant, the variant frequencies are very different in the different places. So you actually have a lot of regression time going back to the mean. Because you wouldn't expect to see a lot of signal if they're already very similar, the frequencies, right? So one thing that's really helpful for each of these variants is that at the initial time, it went through this explosive phase. There's a lot of randomness encoded in space, in the population, which then goes away. And the going away dynamics is what we are looking at. And we are extracting these importation rates from it.
So, well, sorry, could you go back one slide? And that's this, yeah. I'm loud, but there's quite a big increase in incidence sort of over October later on. There's initially, of course, some of that fluctuation is because it's rare too. It's much more less common back in April. But then when you get later on, but there doesn't seem to be, I'm wondering if it's a little surprising that there's relatively little noise arising from what would maybe be stochastic variation in the rate at which different things take off at different places in that time.
Yeah, so as I said, I think this is a mystery to me in general, right? But why is there even a plateau? And we had this discussion.
I'll have an answer. I wonder if it's associated, but we'll talk about that later.
He didn't have an answer for that. He just agreed it's a puzzle. I have different answers also. So. But what would be your answer?
I'm meeting with you later, so I'll tell you then.
Okay.
I don't want to take the time. It's very much to do with networks and the rate with which networks fall off a cliff and become susceptible to being infected. When we talk about networks, you can think of it as clicks, and they don't all do so at the same time. And basically, there's a range at which that happens, and that means that there's a range at which different networks get infected and reinfected, and that produces a plateau, at least in this kind of conditions, unless something changes to the networks, like schools, what have you. But as I say, we can talk about it in more detail later.
Yeah, I'm curious to hear more about that. Anyway, as I said, it was useful for us because it was roughly constant over time. I'm curious whether maybe these exponential sub-outbreaks actually speak or are consistent with what you expect in this period of time then, but we'll talk about it later. No, I wasn't here. Okay, so now there's the mathy part. But it's also relatively simple. So here I'm describing what happens to completely neutral variants over time. How can they be described mathematically? And given that this assumption of neutrality is correct, you can only write down this equation. So there's no approximation here, but it only applies if really your variant is neutral. And that's another question of when is a variant neutral. But so here the discretization of time that we use is 1 week, which is kind of natural. There's it's a natural periodicity of how people got sampled, and also it's approximately the generation time. So our discretization is always a week, and this equation says that the frequency of a given variant in population I in the next week is the same as the frequency in the previous week, apart from two things. One is, of course, demographic noise. That's not the focus of this study, but it's of course there. That's a random change. But then there's another deterministic change, which comes from importations from other groups. And you have to sum over all possible groups J that are not equal to I, from which you can import. And then, for example, if you look at just one J, then you can, due to importation, you can change the frequency if in J the frequency is actually different, right? You can imagine the frequency difference matters. Because if the frequency is the same, then if you import from that region, you don't expect any change. And that's why these differences come in. And then the rest is here a matrix that we write down. And this matrix, we call it the importation rate matrix because that's what it really means. It is the proportion of infections I, region I, imports, deem I imports from deem J per region. We tend to rewrite this equation into a simpler or standard linear form like this, where then we have to define also a diagonal element, which when we do it in this way, the entire matrix becomes what's called a right stochastic matrix, which is useful because then there's another interpretation of this lambda ij, namely lambda ij also represents the rate at which a lineage jumps from population now I to j backward in time. And so how can we understand this? Let me illustrate this forward in time. Of course, you know, if a virus is successful, it generates a tree of descendants. But if you go backward in time, you sample an individual, then then you follow a lineage backward in time, and this lineage can either stay in a deem or jump around. And the jump rate is exactly what lambda ij is. Okay, and then the question is how do we use this framework to infer this matrix? Well, the simplest way would be to just do linear regression. So you just compare the predicted or the expected frequency in the next generation with the actual observed one, and you try to minimize the error over all possible variants. Now, this new index mu is really just a variant index. And that gives you one answer for this matrix. However, that ignores the fact that there is this observational noise. And that's why really a better, you almost have to do this method is a hidden Markov model in which for continuous state space becomes a Kalman filter. So here we say these actual frequencies are not observed. They're kind of hidden variables. And there's a noise between the true variables, true frequencies, and the observed ones. And that can be implemented, and that gives you a posterior distribution then of this matrix. And this is what we use all the time in the results that I'm presenting now. Okay, what do we get out of this? Well, we get a big giant matrix. That's the matrix. Couple of things to say. First is the matrix gets the bigger, the finer resolved I look at England. The most fine-grained resolution you have in this kind of data has a funny name, it's UTLAs, upper tier, local, and then something else I forgot about. But that is turns out a too fine-grained resolution. If we create our matrix on that resolution, you get an answer, but it's too noisy. You can't take it seriously. So there was one important step to think about, what level should we look at England so that we can actually trust these matrices. And so that made us realize we can subdivide England into 50 themes, that was where the threshold is, or less, in order to create a matrix that is meaningful and not too noisy. And so by creating kind of this own subdivision into themes, into 50 themes, we end up with these centroids here, these nodes in England. And so each row and each column corresponds to one of these 50 themes. And then we get this importation matrix lambda ij. And before we really look at this, another thing I have to tell you is we have ordered the different entries here according to a particular way. Namely, we looked at the rows and introduced a distance between locations by looking at the similarity of two rows. If two rows are exactly identical, that would mean that these two regions import from other regions in exactly the same way. Right? So, and then we would say, okay, this distance between the now introduced distance would be very, very small. And so we ordered now these beams in such a way that beams that are next to one another have a relatively small distance. And the distances you can kind of read off from this tree that is generated from clustering. And the first thing that you notice is even though we don't give the computational tool any geographic information, it naturally clusters based on the similarity of these rows, regions or beams together that fall into the same region, one of these 9 regions, right? So gray, for example, is London. They're clustered next to one another, very small distance. Yellow, so you tend to see the same colors next to one another, which is kind of nice. It tells you there's some interesting signal. And also after this clustering, you see approximately here a block structure in this matrix, which means that interactions tend to be stronger. Thank you. Tend to be stronger between nearby beams, which also makes sense. This is also shown here. So here we have drawn a network where we only show a connection. These are all directed connections. We only show a connection if the corresponding entry in the matrix is larger than a threshold. And so in this network, then you see that mostly you kind of import from neighbors, but sometimes there are also long distance connections that are significant. That is also shown here, that inter-regional connections tend to be stronger than intra-regional connections. You can also get here a follow-up of the importation rate with distance. That's shown here on a double log plot, where these are the red points are all the possible connection between two different teams. And this fall-off is here something that we fitted with a power law only to compare it to mobility data, because there some of you might know that from just tracking cell phone data, you can learn something about how people move around, and that tends to be a fall-off, power law fall-off, at least over some distance interval of these mobility interactions. However, in England, we didn't have the mobility data at the time. It was COVID odd period was over, and we couldn't get our hands to the mobility data. So we talked to Moritz Krämer, but he didn't have access anymore at the time. So that's mainly the region I show you now. The corresponding plot for the US, also during the Delta period, we have much less data, and we can only, our deems are much bigger. But I show it nevertheless because here we did have mobility data, and you can now see in blue the fall off in the importation rates that we inferred compared to what we would expect just based on the mobility data, right? So just there's less and less travel, long distance travel with larger distances, you expect some kind of fall off, but this fall off is significantly stronger just based on mobility data than what we infer, which I think is interesting. And you can ask, of course, why might that be? I'm sure there will be different hypotheses. One hypothesis that would make sense to me is, well, let me remind you that these importation rates, they're really a combination of transport and behavior of people and immunity, lots of other things. But it's not just mobility. So you could imagine if you travel a long distance, then you probably have a lot to do locally and talk to people. So you could imagine long distance travel or behave in some sense more risky. So that could be the reason why with distance the decay in these interaction decays more slowly than just based on mobility. There might be other reasons, of course, as well. If you take the airplane, maybe that's risky inherently. Who knows, right? So these hypotheses are always to be taken with a grain of salt. All right, I don't have much more time, so let me be quick here. This just to another way to show that really geography is encoded in this matrix. You can do you can embed this very high dimensional matrix into just a plane using multidimensional scaling. And what pops up is a very crude version of England. London is down here, Northwest is there. It's where there's some geographic information.
Now, clearer versions.
But anyway, this ignores any non-symmetric interactions because the distance is symmetric. And that's, I think, the power of this approach that you can look at asymmetry in importation rates. And so in the next one, we just look at three regions. Here, London is half by east of England and southeast, and we compare it across waves. And so here you see essentially the matrix, a 3x3 matrix drawn as arrows for this period of time. And then for Delta and Omicron, it looks like that. And the main point of this is to show that, well, these matrices change over time. It was a little bit surprising to me. I thought, maybe if you're lucky, you get this matrix and it's just more or less a constant, mainly coined by mobility. Wouldn't that be nice for prediction purposes? But it really does change substantially between waves, less so within waves. So here you see some matrix elements within waves from month to month, how they fluctuate, and these kind of fluctuations are less pronounced. There's some more math here, just associated with the fact that you can look at not only the dynamics from one week to the next, but now that you have that matrix and know that at least in expectation everything is linear, you can also predict the frequency of a variant down the line a couple of weeks later, t weeks later, from the current frequencies by just taking the power of this matrix. And that what that essentially means is that, especially for long times, t, not everything about this matrix matters, but the main principal components matter. That is the reason then for all this math is really just looking at the eigenvalues and eigenvectors of this matrix and, well, realizing it's dominated by the leading ones. What do we learn? Well, the first thing we learn, there are two things you can learn here. One is from this analysis, you can determine something which in evolutionary biology would be called per capita reproductive value. Like how infectious, inherently infectious are different populations. And it turns out we didn't see a lot of difference, at least within waves between the different regions. At least within the error bars, reproductive values were kind of constant. Another thing you learn is about, we call it relaxation time. Essentially, how long does it take for the infection to go around? And that differs not only between waves, but also you can look at the discrepancy between between taking the entire network and then changing the network by severing all the long distance connections. That's at least the kind of little modulation or non-pharmaceutical intervention that we simulated here, we say, okay, let's assume people do not take any long distance transport. What would be the consequence? The consequence would be that indeed the relaxation times would increase. This is actually, anyway, it is correct. So the true relaxation times that we infer here is longer for alpha than for delta, and it is much longer if you ignore these long distance connections. We only have a few of them, and they tend to be quite weak, but if you sever them, then it has a big impact on this relaxation. All right, so with that, I'm really kind of at the end. So we see in correlated fluctuations in cohort importation rates. These importation rates reflect geography, but differ substantially between waves. They are, oh no, this doesn't, we thought we see spatial gradients, but actually we didn't see spatial gradients. So please erase that. I forgot to do that. And then maybe another point here is long range connections really matter more than just expected based on mobility data alone. I have one more thing to say about what I would like to do with this. So we only had labels about locations. If you have other labels like age groups, ethnicities, we can look at their interactions, which would be extremely interesting and potentially important, but we didn't get our hands at that. So if you have any suggestions, please let me know. There are other data of this sort. All you need is time series of neutral variance over time that can be potentially gathered for microbiomes doing metagenomics. And then you could ask, the question, how do microbiomes be imported or shared between different populations, which would be an interesting question. Another thing we're working on is a completely different question, namely using ancient DNA to infer migration patterns going way back past. So, of course, ancient DNA of humans, we have way, way fewer samples than sequenced viruses here in the case that I've discussed. But with human genomes, it's the case that you have recombination that effectively unlinks different parts of the genome. So even though the sample size of complete genomes is much less, you have a very large effective sample size because the genomes, the different regions are unlinked. So different SNPs on different parts of the genome are different, behave roughly independently, at least going sufficiently far back in time. So there's also power there, and you can ask me later if you're interested in that. So with that, let me thank people who did the work. So I already mentioned Chin for especially the first part. She was also involved in the second part. It was then mostly worked on by Takashi really through a couple of years. He did a lot of analysis from different angles. We really double checked everything until we were happy. Now at the end of COVID, we have finally put it on the preprint server. Yeah, and I'd like you to. I'd like to thank you for listening.
Thank you so much. I think we have a few minutes for more questions. Like 5 minutes. Over there. Yeah.
Thank you for the talk. Can you talk a little bit more about the eigenvalues and particularly the interpretability of eigenvalues? Because to my admittedly limited knowledge, PCs are not always great interpretability. Can you talk more about the utility of your eigenvalues?
Yeah, I'm happy to do that. Direction. Then you forced me to go through this math. Well, if you accept that equation, then I can I can always write this vector in term as a sum of eigenvectors with certain coefficients. I can expand a vector in terms of a complete set of basis vectors. And the natural set of basis vectors are the so-called eigenvectors of this matrix, right eigenvectors of this matrix lambda. And we call them here, the right eigenvector is U, and alpha is the index, corresponding index of that eigenvector. And so I write this vector F here in terms of is the component A alpha of that particular eigenvector. And then the nice thing of this equation, you know, if I apply the matrix lambda to an eigenvector U alpha, I just get the same vector multiplied with the eigenvalue lambda alpha. So the right-hand side really just becomes this very, very simple sum over eigenvectors U alpha, and each of these summons contains then the eigenvector to the power of t. And if t becomes large, only one term, or potentially two terms, become really important here. And you can show for a right stochastic matrix, the largest eigenvalue is always 1. So that's what's dominating on long terms. All others will slowly fade away. And the eigenvalue 1 will just lead to a very, very, I mean, we also know what the eigenvector is, just all ones. Which just means that here, neutrally speaking, the frequency, the expected frequency on long terms due to the mixing process will be the same in all deans. That's why the eigenvector has to be 1, 1, 1. But then what tells you about the dynamics is the next to leading eigenvalue. So this is the lambda 1 vector. This is the one that I have plotted here, or the inverse of it, which we interpret as a time. And then, you know, here I haven't said anything about all the sub-leading eigenvalues. About the first eigen value, there's something else to say, namely, even though the right eigenvector is boring all ones, the left eigenvector is interesting. Namely, that tells you something indeed about the reproductive value. It tells you what contribution will a given dean have to the expected long-term mixed population. And that's then represented here. It could have been different. So initially we thought we see a gradient, no self gradient, but there was a bug in the code, and now we see this pattern. Maybe there's some variation, but we also have these error bars. So. Other questions?
Yeah, for a short one.
Okay, I think this is probably short. These are not super recombining genomes, so
Yes.
how do you get more than one effectively SNP, or more than a small number of SNPs that are uncorrelated with each other?
Yeah, so we essentially need lots of low frequency SNPs. So if you imagine, so we have two ways of actually getting at the data, right? One is if we had a full tree for all the sequenced genomes, if we had that, we could just cut very deep in the tree, and then we get independent lineages that we can define and follow. But that requires this tree. An alternative which works extremely well is the one that we then ultimately use, which is a tree-free alternative to that, is we just track all the mutations that we have. If we do that, then the basic mathematical equation, the framework still works. However, you are overly confident in your results because many of these mutations are going to be linked, and so are not really independent. So what we then do is we actually cluster these time series. We introduce also a distance. The more correlated they are, the more they are in a cluster. And then define how many clusters do we want. And for each cluster, we just have one representative. So this is something that Richard Neer used in his work as well. And to just get time series of allele frequencies, and that's what we do here to avoid the use of a tree. But as a control, we have also used a tree that we cut at some point, and we get the same, essentially the same inferences.
You are getting the
Thank you.